Welcome to Global IMMUNOTALK 2024. I'm Kazuyo Moro from Osaka University School of Medicine. We're very excited to have Veronique Angeli for today's speaker, who have done wonderful works on the neutrophils and macrophages. But before moving to her talk, I have two housekeeping announcements. One is about how to make a question. So in this seminar series, we are not going to get the question directly. So please do it through the Twitter. I will explain how to make a question after her talk. And secondly, next week's speaker will be Shabana Kahdi from Washington University. So I hope you can join next week too. So now I'm very happy and honored to introduce Veronique Angeli. She received her PhD in 2001 from University of Lille in France. And she did her postdoctoral training at Mount Sinai School of Medicine in New York City from 2002 to 2004 in the laboratory of Professor Granwan Ladolf, before being recruited as a instructor. And in 2006, she was recruited as assistant professor in National University of Singapore, Yang Du Lin School of Medicine in Department of Microbiology and Immunology, and promoted as a associated professor in 2014. She is currently a leader of the immunology program at the Life Science Institute, NAS, since 2018, and also the director of Immunology Translational Research Program at Yang Lin School of Medicine. And since 2020. And her main research interest focus on the lymphatics, vessel function, and health and disease and tissue resident macrophages. She have done so many wonderful work on the macrophage. And she is currently investigating the pathophysiology of human lipoedema to identify a new therapeutic target and the contribution of Lyve-1 expressing macrophage in the tissue homeostasis. So I think she's going to talk about this today. So now, Veronique, could you please turn on your camera? Okay. Thank you for coming to Global IMMUNOTALK, and we really look forward to have your talk. But before that, we always make one question to know what kind of thought the talk speaker have. And today's question for you is, what is your mentorship style?
Okay, thanks a lot, Kazuya Moro. So yeah, that's an interesting question. I think I would say that I'm quite accommodating. My mentor style is quite accommodating because I feel that you have to adjust to each like student profile. Because maybe one strategy may not work for all, and then secondly also during their career progression from year 1 PhD to year 4 or early postdoc to the end of their postdoc, they might have also different needs. So I try to work together, and really try also to promote their career progression, which I think is also very important aspect, to really discuss, together. And I also try to identify, what are they good at, maybe some might be better at dry labs, than really bench work and so forth. So, we try to tailor a project that suits them as well, And because I think if they have interested, then they will be really productive and have a really great success, for their future career.
Okay, thank you very much.
Sure.
So now, if you are ready, please share us your slide first.
Yes. All right. Can you see my slides?
Perfect.
Yes. Okay.
Okay, so please.
Thank you very much. Okay, so yes, indeed, it's really a great pleasure to be invited today for this Global IMMUNOTALK. I think it's really a great initiative. And so today, I'm going to share our our work on matrix remodeling function of Lyve-1 positive tissue-resident macrophages. But let me indeed just a few words on our research interest in the lab. I think it's really clear that in the past decades that there is emerging evidence that the immune system is really very important to regulate tissue homeostasis, and that extend its function basically beyond its function in a host immune defense. And so we think that a better mechanistic understanding of the regulation of tissue homeostasis by the immune system may also advance the development of strategy for multi-organ disease. And in the lab, we are particularly interested to tackle the regulation of tissue homeostasis by two major components of the immune system. The first one is the lymphatic system, which comprise a network of lymphatics, but also lymph nodes. It's been shown that beyond being important in trafficking of immune cells, induction of immune response, lymphatic vessels are also very important, for example, resolution of inflammation, and also now have been shown to be important in a really diversity of disease, human disease, cardiovascular disease, metabolic disease, neurodegenerative disease, and so forth. So I think we have a lot more to learn about how lymphatics play a role in tissue homeostasis. But today I will focus on the other interest in the lab, which is the importance of tissue-resident macrophages in regulating tissue homeostasis. So let me give you a very brief introduction on macrophages and particularly tissue-resident macrophages and their function. So they were, as probably they were first discovered or described at least by Elie Meshnikov when he was studying very primary animals like a starfish, and showed that there were these cells able basically to ingest like a foreign body, and that really and this process then was later called phagocytosis. So they are really important in a host defense. But like as I mentioned, I think we have increasing evidence that their function go beyond this host defense function, and they are really important in maintaining tissue homeostasis, and they are really integral component of the tissue. In fact, they are present in almost all tissues throughout the development until adulthood. And so what we know about these tissue-resident macrophages is that they are a very heterogeneous cell population, for example, in terms of their origin and also activation state. They also, their function is tissue-specific or niche-specific, and I will share some example today. And they have really a prototypic function in development, health, and disease. And for example, here on this diagram, which from Davis Nature Immunology in 2014, really showing this indeed diverse tissue homeostatic function of macrophages. For example, of course, their importance in clearing erythrocyte in the bone marrow or the spleen. Also, their importance, for example, in resolution of inflammation, maintenance of adipose tissue, either a contraction, for example, of the smooth muscle cells in the intestine, and so forth. So really, really important cells to maintain tissue homeostasis. As I mentioned here, they are really heterogeneous in terms of their origin. So indeed, the dogma that all tissue-resident macrophages arise from basically monocyte-derived cells derived from bone marrow-derived blood monocytes. This has been recently challenged, and this is because of advance in technologies. For example, the use of parabiosis, bone marrow transplantation, and also more recently a lot of fate mapping reporter mice, allowing to identify the origin of these macrophages. And indeed, as shown in this diagram, really tissue-resident macrophages, or TRM, can be derived from both embryonic and monocytic lineage. So during embryogenesis, basically these tissue-resident macrophages will develop from early stage like yolk sac or the fetal liver, and then after a postnatally development, these macrophages are able to either maintain through self-renewal, or they can also be gradually replaced by bone marrow-derived blood monocytes. And this basically will depend on the tissue, basically where this TRM reside. Indeed, for example, if you take tissue-resident macrophages of the brain, also called microglia, they are mainly basically of embryonic lineage. Whereas, for example, in the intestine, you can see that a larger proportion are from monocyte-derived origin and fewer from embryonic origin. So we, in the lab, we were particularly interested about this Lyve-1 expressing macrophages population. So what Lyve-1 stands for? It stands for lymphatic vessel endothelial hyaluronan receptor 1. And so as the name stands, it's a hyaluronic acid receptor or hyaluronan receptor. And in fact, it was primarily known as a lymphatic marker. And as I just mentioned, my lab focused a lot on lymphatic vessel research. And indeed, if you look at this skin whole mount, for example, you can see this beautiful network of lymphatics stained by Lyve-1. However, there were some studies suggesting that macrophage in certain tissue can also, in certain conditions, can also express Lyve-1. And at the time, this was in the early 2000s, there were just few tissue shown to exhibit these Lyve-1 positive macrophages, but now the list of a tissue basically in which Lyve-1 positive macrophages can reside is extended as shown here. However, at the time, the function really of these Lyve-1 positive macrophages was not really known. And this is what motivated us to figure out a little bit more on these Lyve-1 positive macrophages. So now we know also that Lyve-1 macrophages are in fact conserved present in a lot of tissues, for example, the heart, the liver, the lung, the kidney, the brain, but also they are conserved between mouse and human tissue. And this particularly, thanks to the single cell RNA sequencing, showing indeed, this conserved signature of Lyve-1 positive macrophages in mouse and human tissue, so this population expressed TIM4 and particular another tissue-resident macrophage marker for receptor 2. So, like as I mentioned, there is very little known at the time. So this was in maybe 2010 or 15 about particularly also the localization of these Lyve-1 positive macrophages. So what struck us is when we were doing this whole mount skin staining with Lyve-1 is, of course, we detect these beautiful lymphatics, very bright for Lyve-1. And also you can identify these cells that are Lyve-1 and are not lymphatic vessels or vessels. We show that they are indeed macrophages because they express CD68. But what was really surprising is this really kind of train track kind of pattern. It seems like we were lining a structure. And so we did a lot of different staining to try to understand what kind of structure these macrophages could line. And to cut a long story short, we identified that these Lyve-1 positive macrophages line particularly blood vessels, but not all blood vessels. In fact, by staining smooth muscle actin, which stain like for larger vessels covered by smooth muscle cells, such as, for example, your vein, venule, arteries, or arterioles, we can identify that Lyve-1 positive macrophages line the outer layer, basically, of the vessels, but only those that are expressing smooth muscle actin, so that are covered by smooth muscle cells. And this, we show this not only in the mouse skin, but also in human skin. You see these beautiful vessels where the Lyve-1 positive macrophages are basically sitting around these vessels, like in mouse brain as well, vessels, or mouse adipose tissue. So in the mouse, the larger vessels is the aorta. So we study basically these Lyve-1 positive macrophages in the mouse aorta at steady state. Just to, if you're not familiar with the organization or the structure of an artery, the artery is composed of the lumen. Then you have this inner layer called the intima, where it's mainly composed of endothelial cells. Then a layer of called the media, which is mainly composed of elastic fiber, collagen, and very abundant in smooth muscle cells. And then the outer layer is called the adventitia, composed mainly of collagen fibers, a lot of fibroblasts, macrophage, as I will show you soon, and also blood vessels, also called vasovasorum, and lymphatics. So staining just revealed that indeed a very abundant presence of these Lyve-1 positive macrophages on the outer layer of the mouse aorta, as shown in this staining here. And then flow cytometry show that indeed we can identify these tissue-resident macrophages in the aorta, and we observe two populations based on Lyve-1 and MHC class 2 expression, where you can see this Lyve-1 positive MHC class 2 negative population and Lyve-1 positive MHC class 2 positive macrophages. And in fact, when we did a further analysis of the adventitia versus the media and the intima, you can see that the Lyve-1 positive macrophages are the more abundant macrophages compared to the Lyve-1 negative, and this regardless of for both MHC class 2 positive and MHC class 2 negative Lyve-1 positive macrophages. And in human artery, we could also observe these Lyve-1 positive macrophages. So here we had the chance to have some specimen from old individuals and young individuals, and where you can see the media here, the adventitia, and here you can identify this Lyve-1 positive CD68 macrophages in the adventitia of human artery. So with that, we wonder what is the function of these arterial macrophages. And at the time, there was definitely evidence that arterial resident macrophage are present in the steady state, and however, their function, particularly at steady state, was not really well known, in fact. So the first way to approach this question was just to look at the transcriptome of these, comparing the transcriptome of Lyve-1 positive and Lyve-1 negative macrophages, and you can see that indeed they have a very different transcriptome. And when we look at genes that were upregulated by these Lyve-1 negative or Lyve-1 positive macrophages isolated from mouse aorta at steady state, what was really interesting to observe is that these Lyve-1 negative macrophages upregulated genes associated with processes involved more in immune regulation or inflammation. Whereas the Lyve-1 positive macrophages upregulated genes more associated with more homeostatic function, particularly metabolic process, angiogenesis, cardiac muscle tissue growth involving heart morphogenesis, elastic catabolic process, and so forth. So suggesting that maybe these two types of macrophage may have very different function. So to basically address, the function of these, specifically of these Lyve-1 positive macrophages, we developed a mouse model in which we can deplete specifically these Lyve-1 positive macrophages. And we show that the Lyve-1 positive macrophages that are present on the adventitia of a mouse aorta express a high level of CSF1 receptor, in fact, higher than level than the Lyve-1 negative macrophages. And as you probably know, CSF1 is a key factor, growth factor for macrophages, and also to sustain their survival. So we decided to cross Lyve-1 Cree with CSF1 receptor LOXP mice to generate a mouse in which basically basically Lyve-1 promoter, we can basically here we can deplete specifically CSF1 receptor in cells that express Lyve-1. So in majority, like the macrophage, but because lymphatics are not expressing CSF1 receptor. So we show that indeed this mouse model is efficient to deplete specifically the Lyve-1 positive macrophages, and this regardless whether they're MHC class 2 negative or MHC class 2 positive. And very important is that the Lyve-1 negative macrophages, which is a minor population in the mouse aorta, is not affected. And we also show that, in fact, monocytes, whether in the aorta or the blood, were not affected in these mice. So we have a nice model now to see what might be the function of these macrophages, see what happened after depletion of this macrophage. So what we observe is like a significant remodeling of the mouse aorta at steady state in these mice lacking Lyve-1 positive macrophages. Particularly, we see increase in the adventitia area, increase in luminal diameter. And so when we did some collagen staining, either specific for collagen type 1 or serous red, we stain all collagen, we observe really an accumulation of this collagen in the media and also in the adventitia layer as shown in both staining here. And this was also revealed by quantification of this serous red in the mouse aorta. So where you can see definitely an increase and accumulation of this collagen in absence of these Lyve-1 positive macrophages. So collagen is known to control the function and particularly the vascular tone of the vessels. So we decided to measure basically the stiffening of these vessels. And it's because it's also known that collagen accumulation can induce the stiffening of vessels. And indeed, by looking at the biomechanical properties of the vessels, we can see that in absence of Lyve-1 positive macrophages, we have an increased stiffness of the vessels. So here's suggesting first that the Lyve-1 positive macrophages are important basically for the homeostasis of arteries, and particularly by preventing this collagen expression accumulation in the vessels. So then we venture into deciphering the mechanism underlying basically this function of Lyve-1 positive macrophages. And as the macrophages basically are present in adventitia, and we noticed that they were really in close contact with the smooth muscle cells present in the media. And in fact, the smooth muscle cells are known to be one of the major producer of collagen, elastin in the arterial wall. So we wonder whether there is basically the special interaction between the macrophage and the smooth muscle cells, and whether these macrophage could regulate the production of collagen by smooth muscle cells. So we went on by doing a co-culture system here, where we sorted macrophages and we cultured them with primary mouse aortic smooth muscle cells. And then 24 hours later, we basically measure the collagen protein expression by smooth muscle cells. So you can see these are the non-treated cells, smooth muscle cells. We express some collagen, but what was really striking is that when you add Lyve-1 positive macrophages, you can see a decrease in this collagen, whereas the Lyve-1 negative macrophages were not able to do have such effect. And this is quantified here, where definitely your Lyve-1 positive macrophages can basically decrease the collagen, the amount of collagen produced by smooth muscle cells. So we try to understand what be the mechanism, and in fact, we thought that maybe these Lyve-1 positive macrophages may regulate the synthesis of collagen by smooth muscle cells, but it was not the case. So we hypothesized that maybe they are able to basically control this collagen expression by smooth muscle cells more at the protein level, and particularly through degradation. And notably, there was some data showing the importance of this matrix metalloproteinase 9, MMP9, in controlling arterial stiffening. And also our data show that Lyve-1 positive macrophages express higher level of MMP9 gene compared to Lyve-1 negative macrophages. And indeed, by staining, we can see also that these Lyve-1 positive macrophages express MMP9. And we also show that using in situ thermography, that MMP's activity was significantly decreased in absence of these Lyve-1 positive macrophages. So this suggests that maybe this mechanism might be dependent on MMP9. So we did several experiments here. First, we, for example, use a MMP9 inhibitor in this co-culture system. So you can see here where when we have compared to Lyve-1 positive culture with smooth muscle cells, when you add the inhibitor, basically you abrogate this particularly regulation of collagen expression induced by Lyve-1 positive macrophages. And this, if you have the smooth muscle cells with MMP9 inhibitor, it's not affected. So suggesting really that these Lyve-1 positive macrophages regulate collagen production via MMP9 dependent pathway. And to further confirm these data, we also in our co-culture system here, compare wild-type Lyve-1 macrophages or Lyve-1 macrophages which lack MMP9 expression. And you can indeed show that in this case, in absence of MMP9, these Lyve-1 positive macrophages are not able basically to control this collagen production by smooth muscle cells. So here really suggesting that this control of collagen production is required MMP9 dependent proteolysis. So it's really through degradation of collagen. And so we wonder whether this interaction between the smooth muscle cells and the Lyve-1 positive macrophages require cell contact or not. And because there were also very interesting paper, and particularly in the cancer context, showing that MMP9 is not just secreted, but can be expressed pericellularly. And so we wonder whether this might be also the case in our basically system here. And so what we did, it's just basically try to mimic the effect of Lyve-1 positive macrophages by adding pro-MMP9 or active MMP9 to the smooth muscle cells. But as you can see here, there is no really differences in terms of collagen production in these conditions. So suggesting that just adding this MMP9 doesn't mimic the effect of this Lyve-1 positive macrophages. So really suggesting that maybe you need cell-cell interaction. And to really further confirm this concept, basically, we then use an assay where the two cells were separated through do a transwell, and as you can see here, only when the cells are in contact, you can see this basically regulation of collagen type 1 expression by the smooth vascular cells, and not when the cells are separated. So meaning that you really need this interaction between the smooth vascular cells and the Lyve-1, and suggesting that indeed MMP9 might be basically expressed cellularly, at the surface of the Lyve-1 that can basically engage the collagen produced by smooth vascular cells. So to further dissect this mechanism, and particularly try to understand what might be the receptor involved in the pericellular localization of this MMP9 on Lyve-1 positive macrophages, we did a series of experiments where, based on literature, it was shown that this MMP9, for example, can localize peripherally via binding of to a few receptors, for example, CD44, and also integrin beta-1. So we, in our co-culture system here, between smooth vascular cells and Lyve-1, we have a condition where we add this blocking antibody against CD44 or integrin beta-1. But you can see that this did not affect basically the degradation of collagen type 1 by the Lyve-1 positive macrophages. And of course, we realized that CD44 is a hyaluronan receptor like Lyve-1. So this suggested to us that maybe Lyve-1 per se is not just a marker of these Lyve-1 positive macrophages, but might play a role in really mediating the function of these macrophages. So we went on to use several blocking antibodies specifically against Lyve-1, and indeed we showed that both of these blocking antibody basically alter the function of these Lyve-1 positive macrophages, and so they are not able anymore to degrade these collagen type 1 produced by smooth vascular cells. So suggesting that Lyve-1 is important in this cell-cell interaction. As I mentioned, Lyve-1 is a receptor for hyaluronan or hyaluronic acid. So to test whether indeed the engagement of Lyve-1 and HA is important to mediate Lyve-1 positive macrophages function, we then went on to use an enzyme that is known to degrade HA produced by the smooth vascular cells. So we use hyaluronidase. And you can see these are the smooth vascular cells producing this nice HA stained by hyaluronic binding protein, and when you treat the cells with hyaluronidase, then you can really see the decrease in HA. And indeed, when you have Lyve-1 positive macrophages which were in contact with smooth vascular cells treated with hyaluronidase, then these abrogate their effect on collagen type 1 expression. So this story basically was published in 2018, and here is the summary of that study. So what we found is that really these Lyve-1 positive macrophages are really important to maintain arterial homeostasis. So they are present in the adventitia, they are in close contact with smooth vascular cells, and which engage basically to the receptor Lyve-1 at the surface of your macrophages, which engage HA produced by smooth vascular cells. So this brings the cells in close contact, and the Lyve-1 positive macrophages produce MMP9 pericellularly, which is able to degrade basically the collagen produced by smooth vascular cells. When there is an absence of this macrophage, as a consequence, you see an accumulation of this collagen in the arterial wall, in the media, and the adventitia, and this induces basically arterial stiffening. So really showing the importance of these macrophages through the regulation of collagen content, like in maintaining basically arterial homeostasis, and particularly preventing arterial stiffening. Since then, we have more evidence that this control of collagen content is not restricted to the aorta, In fact, in that study, we also show that in other organs, like for example, esophagus, in absence of this macrophage, you can see really this deposition of abnormal deposition of collagen. And this was quantified here, and also in collaboration with Florent Ginoux, who is now with Gustave Roussy in Paris. We show that indeed there is a population of Lyve-1 positive macrophages in the lung, which are also very important to limit the exacerbation of collagen deposition in bleomycin model, so a fibrotic model. So really showing that, the role of these Lyve-1 positive macrophages in collagen, regulation might not be limited to the aorta. And in fact, we have an ongoing study also showing the importance of this regulation in the skin. And maybe next year to another global IMMUNOTALK, I may share that study with you. But now I like to focus to an unpublished, in fact, study, which is a follow-up basically on that first, study. Having shown that these Lyve-1 positive macrophages are indeed very important, in the arteries, and particularly maintaining, arterial homeostasis. Of course, we, naturally looking at now like their potential role in arterial diseases, such as, for example, atherosclerosis, which are characterized by also a remodeling of the arterial wall, and particularly, lipid accumulation, but also extracellular matrix. Aneurysm, which is like a disease where you have dilatation of the aorta and then rupture, and is also involving a lot of ECM remodeling. And today, what I would like to share is the study on the Lyve-1 positive macrophages in the context of vascular aging. And this is, in fact, is the study by a very talented postdoc in the lab, Xiaowing. So indeed, arterial stiffening is one of the hallmark of vascular aging, and is known to be one of the major risk factor, in fact, for cardiovascular disease. So during aging, indeed, it's not just your skin that change, but your arteries are also affected. And as you can show here on this diagram, there's a lot of remodeling of these arteries with age. Characterized by endothelial dysfunction. There is also particularly vascular stiffening, and this is particularly related to the fact that you have also increased fibrosis or accumulation of this collagen, and over aberrant extracellular matrix, you have inflammation. Changes in the media layer, and characterized by thickening of the layer, but also because of proliferation of these smooth muscle cells. So all these will lead to basically an increased vascular diameter overall, or also increased diameter in the adventitia and the media layer. So we wonder, since we show that they are critical to maintain vascular homeostasis at steady state, what happened to these cells during aging, and can they maybe contribute to this arterial stiffening observed during aging? So to do that, we study 2-year-old mouse, because as shown here, they show basically all marks of this vascular aging. For example, increase in medial thickness, increase in luminal diameter. Also, they show this arterial stiffening measured by pulse wave velocity, basically the ultrasound method, which is in fact used also in the clinic to look at arterial stiffening in humans. And as well as I mentioned, vascular aging is associated with this collagen accumulation, and you can see indeed media collagen accumulation, and this is really clear also by this staining for collagen type 1, which accumulate in the media and in the adventitia, in particularly in these old wild type mice. So using what we call young, so means like 20 weeks of age mice and 2 years old mice. We first went to look at more article cell heterogeneity like during aging. So we sought cells, myeloid cells and stromal cells, and then proceed with single cell RNA sequencing. You can see really great cell heterogeneity with basically abundance of endothelial cells, stroma cells, fibroblasts in particular, but also macrophages. When we look at macrophages, we could see several clusters of macrophages. And you can see that they are affected, the frequency of these macrophages is affected during aging. And in particular, when we look at genes associated with its different cluster, we can observe 0 and 6 cluster are basically the Lyve-1 positive macrophages that correspond to those that express MHC class II gene or not. And the rest of the macrophage are more like an inflammatory macrophage. And you can see that, in fact, these inflammatory macrophages increase with aging. And there are some changes in this Lyve-1 positive macrophages cluster. So this, then we went on to do a bit more flow cytometry analysis to look at indeed the dynamics of these macrophages in young and old mice. And you can see like we see a decrease in frequency of these Lyve-1 positive macrophages, whereas we see an increased frequency of Lyve-1 negative macrophages. And remember, those are usually a bit more associated with immune response or inflammation. And although with age, we see an increased number of, in general, hematopoietic cells in the aorta in 2 years old mice, you can see that the ratio between Lyve-1 positive and Lyve-1 negative macrophages decrease. So suggesting it's because you have proportionally a greater increase into Lyve-1 negative macrophages compared to the Lyve-1 positive. And suggesting that we may, this is in fact in line with, aging is associated with a mild inflammation, although there is not infection, so it's sterile inflammation. And this is supported by also, an increase in blood monocytes and also in aortic monocytes. So really showing, that there is definitely inflammation in these old arteries. And we made a similar observation in human age arteries. So we had the chance to have specimens from individuals that are below 50 years of age and those that are above 50 years of age. Usually this is the age where, a lot of aging associated disease occur. And indeed, as I've shown you earlier, usually we see these nice Lyve-1 positive macrophages in the adventitia, but you can see that their basically number is significantly reduced in older individuals. And this is represented here, where again, like in a mouse, we see this change in ratio of Lyve-1 positive versus Lyve-1 negative macrophages, because proportionally there is a greater increase in Lyve-1 negative macrophages. So this, of course, prompted us to test whether these old Lyve-1 macrophages are they still able to regulate like collagen basically expression by the smooth master cells using our co-culture system. So here we sorted macrophages either from young, so 20 weeks of age, or old mice, 2 years old mice, and culture them with primary mouse aortic smooth master cells. So in this case, the smooth master cells were from the young mouse, and then we look at 24 hours later. And you can see that, as I shown you earlier, the young Lyve-1 macrophages are able to indeed control this collagen type 1 expression. However, when they are old, they lose this capacity basically to control collagen production by smooth master cells. So suggesting that, they really lose this capacity. As I mentioned in my introduction, tissue-resident macrophages, and including Lyve-1 positive macrophages, they can be derived either from blood monocytes, bone marrow blood monocytes, or like embryonic lineage. And so we wonder whether the function of these Lyve-1 positive macrophages is similarly affected, whether they are monocytes derived or embryonic derived. The first approach we use is this parabiosis, so where basically two mice are linked together and they share the blood circulation. And this has allowed, in fact, to look at the origin of cells in basically the recipient mice, and to see the contribution of the donor cells to these recipient cells. So we look at, here we have, of course, young to old controls, and then also young to old mice. But in this case, so we are looking at mice 6 or 12 months later. And what we found is that this is the data here. So you can see that, of course, the blood monocytes are a positive control because they are replaced and from basically the donor origin. But Lyve-1 positive, Lyve-1 negative macrophages, you see there is like only a small replacement at 6 months, but at 1 year, all the Lyve-1 negative macrophages were replaced. But this was basically not the case for the Lyve-1 positive macrophage, where they are basically remaining still basically from embryonic derived, but also like monocyte derived. So when we sought basically these Lyve-1 macrophages from either the host or the donor and test their capacity to regulate collagen, you can see that regardless they are monocytes derived or embryonic derived, they are not basically able to degrade this collagen unlike the young macrophages. And to further support this data and to confirm, we use another model, the MS4A3 model, which is a reporter mice where MS4A3 is a gene that is be able basically to track monocytes derived cells. So 100% of monocytes will be positive, and then so you can distinguish the embryonic versus the monocyte derived macrophages. And you can see, like for parabiosis, the Lyve-1 negative macrophages over time are basically replaced by the monocytes, whereas like we have more a mix for the Lyve-1 positive macrophages. And when we look at basically, we sort specifically MSF4 positive, which are monocyte derived, versus the MSF4 negative, and test the capacity of degrading collagen expression, degradation of collagen, you can see that basically both are altered when these cells are old, basically. So suggesting that whether monocyte or embryonic derived Lyve-1 positive macrophages, they are both like aging similarly and losing this capacity basically of degrading collagen produced by smooth muscle cells. So what could be, explaining that these Lyve-1 positive macrophages lose their capacity to degrade collagen? And so in fact, we show that MMP9 expression is not affected. So what was like quite interesting is that in a young mouse, as I showed you earlier, these Lyve-1 positive macrophages are in close contact with like the media in particular. But when the mice are old, you can see that these Lyve-1 macrophages are much more distant away from the media, suggesting that maybe there is an alteration in this crosstalk between these Lyve-1 positive macrophages and the smooth muscle cells, which I showed you is very important to mediate their function. Indeed, so you can see here they are much more distant away, in the old mice compared to the young. So to test this, we here did another experiment co-culture system where instead of having the smooth muscle cells, young smooth muscle cells, we compare with old smooth muscle cells. But in this case, the Lyve-1 positive macrophages were young. You can see the young and the old smooth muscle cells express similar level of collagen. However, when we add, of course, the Lyve-1 positive macrophages young into young, we have that degradation or like regulation of collagen type 1 expression by the Lyve-1. But however, this now doesn't happen if your smooth muscle cells are old. So suggesting that indeed, if there is an alteration between this interaction between smooth muscle cells and Lyve-1, this will have an impact on their function. And so we look into this alteration, what could explain, these changes in this interaction between the two cell type. And of course, we look at Lyve-1 expression, but we didn't see any changes in Lyve-1 expression on macrophages, whether they are young or old. And so we look into more, the smooth muscle cells, and particularly whether there is any changes in HA content in aorta. And in a young mouse, we can see very abundant presence of HA. Staying here with HA binding protein, particularly in the media, but also really in adventitia. And this is significantly decreased in the old mice. And this was also further observed in the human artery, where you can see indeed a really significant decrease of HA expression in the adventitia and the media in artery from elderly. And what was interesting also, we noticed in our co-culture experiment that when, in the presence of smooth muscle cells and Lyve-1 that are from young mouse, we noticed also that the Lyve-1 positive macrophages really induce this particular pericellular expression of HA, which is not observed when the macrophages are old. And this is also represented here, where overall the Lyve-1 positive macrophages basically are able to induce HA expression by the smooth muscle cells. But when they are old, they are not able to do so. And also to induce this particular HA pericellular coat on the smooth muscle cells. So suggesting that indeed, you need these Lyve-1 positive macrophages, like young Lyve-1 macrophages, to also induce this HA expression on the smooth muscle cells. And we think that maybe during aging, this interaction, is altered, and this will lead also to changes in the smooth muscle cells, which are not able to engage with the Lyve-1 macrophages. And indeed, when we did bulk RNA sequencing of smooth muscle cells isolated from young versus old, you can see that there's dramatic changes with age. And when we look at also our single cell RNA sequencing, what was interesting to note is that we see when we look at genes upregulated in young smooth muscle cells, you can see that these genes are more associated with, for example, ECM organization, artery morphogenesis, really different metabolic processes involved in ECM, or cardiac functions and so forth. Whereas in the old smooth muscle cells, genes that were upregulated are more associated with inflammation, regulation of immune functions, and so forth. So really showing that alteration of this crosstalk may alter both the Lyve-1 positive macrophages as well as the smooth muscle cells, and that maintaining this crosstalk between the cells is really important to prevent arterial stiffening. So, of course, now the big question was whether we can rejuvenate these arterial Lyve-1 positive macrophages to reverse arterial stiffening. And so there are, as you may know, several strategies now that are employed to look at rejuvenation of tissue, for example, the liver, the bone, the muscle, the heart. And for example, some strategies are based more on introducing young blood factors either by injection of blood factors or parabiosis that I show you. Also ablation of senescent cells, which might also contribute to, for example, inflammation and alteration of these organs. Cellular reprogramming, and also there's a lot of evidence that metabolic manipulation, and particularly the inhibition of this mTOR pathway using rapamycin, has been shown to basically rejuvenate several organs, including the vasculature. Indeed, rapamycin has been shown to basically improve vascular aging. So we decided to employ this strategy and to see whether this improvement in vascular aging induced by rapamycin could be mediated by this reversing basically or ameliorating the Lyve-1 positive macrophages function. So we have here wild-type mice, or also our Lyve-1 depleted mice, so the model that I presented earlier. And when they were 21 months old, they were either put on a control diet for 3 months or a diet containing rapamycin, the inhibitor of mTOR. And in fact, we show that in the aorta, mTOR signaling is significantly increased. So as shown in the literature, rapamycin can basically improve vascular aging. So if you look at these two first control here, control, non-treated or treated with rapamycin in the wild-type mice, you can see that indeed now adenocellular area decrease, media area, media thickness, as well as also the collagen content. However, this basically reversing effect of rapamycin was not observed when the mice lack these Lyve-1 positive macrophages. So really suggesting that these Lyve-1 positive macrophages are very important to mediate basically the reversal of vascular aging induced by rapamycin, and really showing the importance of these macrophages in basically preventing basically arterial stiffening during aging. So of course this experiment prompted us to look at the function of these Lyve-1 macrophages, and we also noticed by immunostaining that now these cells remember, so this is like control mice, usually they are close to the media, but they are a bit distant away. When we treat it with rapamycin, they are more close. So that's here. And then also for function, we can observe that indeed, when we isolate macrophages, Lyve-1 macrophages from mice that were treated with rapamycin, compared to old Lyve-1 positive macrophages, now we can basically reverse and basically ameliorate their function in terms of collagen regulation. So we can, using rapamycin, we can rejuvenate these cells. So we then look at a bit more in details, like what could be basically the effect of this rapamycin and how it rejuvenates these Lyve-1 positive macrophages. In fact, when we look at our single-cell RNA sequencing, where here we compare young versus old and old mice treated with rapamycin, you can see that in these macrophages, we see really some changes in terms of genes that are with different functions. So in the young, they are associated with express more genes associated with homeostatic function compared to the old, which are more immune-related. And then this is somehow reverse in the after treatment with rapamycin. So really showing that rapamycin affects the phenotype of these macrophages. And not surprisingly, when we look at these, senescence-associated secretory phenotype, we can also see this signature in the old mice, and rapamycin kind of reverse some of the expression of these genes associated with SASP. For example, here are some examples of this senescence-associated secretory phenotype. where you can see it is indeed, like a change when we treat with rapamycin. These are key senescence marker, the P21 and P16, where again there is some changes in these after treatment with rapamycin. And what was also interesting to note is that with age, these Lyve-1 positive macrophages have a more pro-inflammatory phenotype, which is in line with the single cells RNA sequencing here. When we look at the bulk RNA sequencing, you can indeed see that the old express a lot of inflammation-related genes, and this is basically partially reversed upon treatment with rapamycin. So really showing that by rapamycin, so inhibiting the mTOR signaling, we basically affect the phenotype of this macrophage, where you have more senescence associatory phenotype and more pro-inflammatory, and now more like looking at acquiring more phenotype of young, basically mice. And also, we look at like this crosstalk between the Lyve-1 macrophage and the smooth mast cells. And when basically these Lyve-1 positive macrophages are rejuvenating, you can see that also we have an increased expression of HA. And this was also measured by measuring HA protein in the aorta, where you can see that in the old mice, you have really a decrease in the abundance of this HA, which is basically reversed by treatment of rapamycin. And the smooth mast cells are also basically their phenotype, which is also a bit more pro-inflammatory, and senescence is also basically ameliorated after rapamycin treatment. So with that, these are the message for this last part, where I hope to convince you that Lyve-1 positive macrophages are really important, in maintaining the homeostasis of the arteries, and this is through the control of extracellular matrix. The malfunction can contribute to arterial stiffening associated with aging. And I think our work really showed the importance of this crosstalk between Lyve-1 positive macrophages and smooth mast cells to maintain our artery healthy. And this population indeed could be maybe a biomarker of healthy vasculature. But overall, because of our data in over tissue, as I've mentioned, in the lung, esophagus, and now in the skin, and over people have shown their importance also in the brain, for example. And this was recently published by the group of Jonathan Kipnis in Washington University. We like to propose that, these really tissue-resistant Lyve-1 positive macrophages as a master regulator of tissue matrix through the crosstalk with stroma cells. And we hope, we can contribute to this particular field. And with that, I'd like to finish by acknowledging the people involved in the work and our collaborators, so particularly in the lab, Angeline and Xiaowing, who really were the co-first author of the immunity paper, and then now Xiaowing continuing on the study on aging. But this, won't be possible without the support of all the members in the lab. And then our funding agency from Singapore, so the National Medical Research Council, NRF, National Research Foundation, and Ministry of Education. And of course, our collaborator from overseas, particularly Florent, who shared with us a lot of different tools, particularly mouse, the reporter mouse, to track like monocyte-derived and embryonic-derived Lyve-1 macrophages. David Jackson from University of Oxford, who is the father of Lyve-1 discovery. Greg Jones for providing us the human samples, and a lot of collaborator from Singapore, as well as our core facility. And with that, I would like to thank you. I hope maybe to see you in Singapore one day. And with that, I will be happy to answer a question later on via Twitter. Thank you very much.
Thank you, Veronique. It was really wonderful talk, and I'm so sorry that we don't have a time for the question. Yeah, but thank you again. And before closing, I would like to share one slide. Mm. Yes, so if you have any question to Veronique, please do it through the Twitter. And first, you need to search for the account Global IMMUNOTALK and find tweet that say ask question for Dr. Veronique Angeli here. And then reply to that Twitter with your question. And please don't forget to mention hashtag Global Immun. So that's all for today and have a nice day. Thank you very much.